Hello everyone, welcome back to another class on regeneration biology. And in today's class, we will learn about the dynamics of immune system in organ regeneration. Like in the previous class, we learned about the involvement of T-Rex, and in today's class, we will learn a lot more about the T-Rex in a regeneration point of view. And it should be clearly understood now that regeneration is not a simple cellular or tissue level event. It also depends on the physiology of the animal, and it also depends on how efficiently the animal or that given tissue of that animal is able to deal with immune cells. So this is very important to follow it up. Let us see some examples. Examples can be from different species, different animal. Zebrafish can efficiently regenerate complex tissue structures with highly developed innate and adaptive immune system. Zebrafish has got a perfect immune system to deal with all this scenario, which provides a model to investigate the roles of immune system or immune cells during tissue repair and regeneration. So zebrafish not only has that regenerative capacity, but it has got a well developed immune system. In other words, if you find some uniqueness in mammal in immune system, you will automatically find a homologue. Say you are talking about T-Rex cells, you are talking about granulocytes, or you are talking about monocytes. You have similar cells in zebrafish, so that makes it an attractive organism to explore in the immune angle as well. Two research groups have reported zebrafish mutants that is deficient in forehead box P3 or FOXP3 ortholog, which helped to reveal the conserved immunosuppressive function of zebrafish FOXP3 in vivo. So FOXP3 is a functional marker of T-Rex, or in other words, if FOXP3 is missing, regulatory T cells are useless or they are not functionless. It's almost like if your hand is missing, you are there as normal. Your leg is missing, you are there. What about if your head is missing? Your identity is lost, life also is lost. So same way, FOXP3 is an important gene for regulatory T cells. Zebrafish FOXP3 defines the development of a subset of T cell lineage with conserved gene expression profile of mammalian regulatory T cells. Like I told you some time ago, that zebrafish have lot in common to that of mammals. In damaged organs, zebrafish T-Rex rapidly migrate to the injury site, promoting the proliferation of the regeneration or regenerative precursor cells by producing tissue-specific regenerative factors through a distinct mechanism from the canonical anti-inflammatory pathway. So T-Rex cells are essential to be present in the injured site if you want to have a proper regenerative response. So these lines of findings illuminate the potential for using zebrafish as an effective model in T-Reg research, focusing on the regulatory T cells, and demonstrate organ-specific roles of T-Rex in maintaining pro-regenerative capacity that could potentially be harnessed for use in diverse regenerative scenario or regenerative therapies. So if we look further into the background of the T-Rex, they play essential roles in maintaining tolerance to self-antigens. Like your body do not produce antibody against your own tissues or proteins. If it do, then that is called autoimmune disease. If any of your organs are now target of your own immune system, that organ will be damaged, and this T-Rex play a major role. T-Rex helps to prevent autoimmune disease and restrain aberrant or excessive immune response to non-self antigens. Self antigen means your own proteins, non-self antigens and at the inflammation site. So excessive immune response is always problematic. Probably during COVID season, you may have heard that many of the dead patients' bloodstream had high levels of IL-6, interleukin 6, which can trigger a very pro-inflammatory environment, and also it can cause pneumonia, etc. So it is important to note that the Tregs allow adequate inflammatory response even if there is a non-self antigen. That means a bacteria or a virus, etc. So Tregs development and function are controlled by master regulatory gene that encodes FOXP3 protein in mammals. Like I told you, Tregs are Tregs because of FOXP3. And the deficiency in FOXP3 function causes fatal autoimmune syndrome both in mice and human. If FOXP3 gene is mutated, both mice and humans will develop autoimmune disease. Treg mediated immunosuppressive mechanisms have been extensively investigated in diverse immunological contexts, but the pleiotropic function of Tregs has been increasingly recognized in recent research studies. So in certain mouse tissues, Tregs infiltrate at the wound site. Okay, so they are welcome. They have to come in the wound site, providing canonical immunosuppressive function and paracrine signals that promote the proliferation and differentiation of resident tissue stem cell. So the Tregs has to come to favor this pro-regenerative environment. And these resident stem cells, their proliferation, etc., because of the paracrine roles played by the Tregs, it will facilitate efficient repair or regeneration of damaged tissues. You may have noticed, even if you get a small scratch wound, if your health is not fine, that means you are not been sleeping properly for several days, etc., your immune system gets affected and that site remain inflamed for a long time. But if you are healthy and perfect, it heals in no time. Simple wound healing, not about regeneration. So the discovery of regenerative function of Tregs led to an interesting question in regeneration biology, whether Tregs exist in animals with robust regenerative capacity, like zebrafish. Does it have the Tregs? Such as urodil amphibians, such as axolotl, newt, etc. If so, to what extent Tregs contribute to organ regeneration? What is their say? Can we address in that angle? So now researchers did in vivo characterization of the function of zebrafish FOXP3. Two FOXP3 orthologs are there in zebrafish, FOXP3A and FOXP3B. Remember, FOXP3 is essential for Tregs formation, function, development, everything, and even the routine function. And they are found to be present in the zebrafish genome 3A and 3B, FOXP3 and 3B, and could play a redundant role. That is the assumption, because teleost fish have undergone some tetraploidization event around 340 million years ago. So this tetraploidization made a diploid organism out of it. Say if our haploid genome is 23 pairs of chromosome, diploid is 46, and 46 becomes, after division, it becomes 92 in the S phase of the cell cycle. And now what if 92 became your 2n? Then you can tell that you got tetraploidized. So your haploid will be 46. Instead of 23, your haploid genome will be 46 now, if 96 become normalized. So that is called tetraploidization. So then what will happen? 46 is a doubling of 23. So 23 had your entire genome. Now it became 46. So you will have a duplication of entire genes. So that has happened for teleost fishes. That's why they have got duplication of many of these genes. FOXP3 function in immunosuppressive machinery is evolutionarily conserved in non-mammalian vertebrates, and FOXP3 plays an immunomodulatory role in zebrafish as well. Mutant zebrafish lines which has got loss of function FOXP3A. There are mutants available where the FOXP3 gene is mutated. They don't have FOXP3A, not B, A alleles. They are used for assessing inflammatory responses in immune cell homeostasis in zebrafish. So homozygous mutant zebrafish, that means FOXP3A is missing. 3B is there, 3A is missing. Zebrafish are completely normal during their juvenile stages but exhibit profound infiltration of mononuclear cells in the peripheral tissue with age. Peripheral tissue means outside the boundary of the cell, boundary of the body. With age, including skin, gills, mucosal tissues of the pharynx and intestine, and subsequent gradual decrease in the growth and survival. So we can see this in this picture. So analysis of FOXP3A mutant in zebrafish. So this is wild type and this is the mutant. We know this is 95-day-old. That means around 3 months old. They are adult, but this is normal size wild type. This is little smaller. So we can see FOXP3 wild type, you have normal amount of in gill, skin and pharynx, you have normal supply of mononuclear cells, but whereas FOXP3A mutant here, you have too much of cell infiltration. You can see here number of cells are way too high. So the inflammatory cell infiltration is seen in Treg depleted or the FOXP3 depleted cell. The mononuclear cells are super abundant without any injury, without for no reason, and they make it inflamed. The environment is now kept inflammatory present, and this is seen with HE staining. And these mutants are appearing normal, but they are highly inflamed scenario. So homozygous zebrafish mutant are entirely normal during their early stages of development, but the infiltration of mononuclear cells are seen across all peripheral tissues. And with age, as the animal gets older and older, there is too much of infiltration of the mucosal tissues are being seen, like skin, pharynx, and also gills, etc. And there is also, this causes also a gradual decrease in the growth of the animal itself. Now, zebrafish Treg function and its role in organ regeneration. Zebrafish effectively regenerate the heart through the proliferation of the existing cardiomyocytes. We all know if you look into the heart regeneration. FOXP3A positive cells, FOXP3A expressing cells actively infiltrate the damaged heart from 3 to 7 days after injury. That means Treg cells 3 to 7 days after injury and remain in the damaged myocardium until the regeneration is completed. The peripheral blood, on the other hand, you are looking into peripheral blood. That means heart is injured. Now we are looking into the animal's blood, zebrafish blood. FOXP3A positive cells are increased by 10-fold after an injury, explaining that the mobilization of FOXP3A cells to the injured site is just normal. Why you found too much of cells from 3 to 7 days in the heart, injured heart? Because it is very highly present in the blood itself because of the injury. Of course, the injury of the heart has triggered this increase. Otherwise, it will not be present. These are all in normal cell. Okay. In the wounded area, FOXP3A positive cells are found within the vicinity of proliferating cardiomyocyte cells, not somewhere else. It is right in the place where the proliferation is occurring. With some of the Treg cells are found to be directly associated with the proliferating cell, which suggests a paracrine role in the myocardial regeneration. So this FOXP3 cells migrated into the heart. Anyway, it is present highly in the blood and migrated into the heart favors the cardiomyocytes proliferation. Sometimes it is very much close proximity, suggesting that the proliferation is influenced by the paracrine function of the Treg cells. After cardiac damage, zebrafish Tregs ingress from the bloodstream into the regenerating myocardium, where the mature Tregs are activated and expand as opposed to the differentiation of the peripheral Tregs in the injury site. They migrate from the bloodstream and expand in number. This is what usually happens. So in this picture, you can see the tissue-specific function of zebrafish Tregs. So sections of injured and uninjured FOXP3A back tag RFP tissue. This is a transgenic line used for detecting the Treg cells physically. You can see no injury and after injury, cardiac injury. And what you are seeing is this is a gel picture of various genes like NTF, NRG, IGF, IL-10. Different genes are being looked in different tissues. They have looked for kidney cells, spinal cord, heart and retina. They have looked for what happens in the normal fish as well as Treg depleted or FOXP3 mutant fishes. And here we can see there is also the control is different actin is used as a control, and also immunofluorescence has been done in the heart for the myosin heavy chain marks which is seen in the cardiac muscle, and asterisks indicate the center of the injury spot, and the dashed line indicates the outline of the injury in these pictures what you are seeing. And in panel B what you are seeing is the picromegaly staining of the heart sections from control left and the Treg depleted right, that is FOXP3 mutant, which is you are seeing here. And C, its zebrafish Tregs are purified from the injured and the uninjured kidneys as control and analyzed by qPCR, and various genes have been looked for, and they have used whether the Tregs are migrating to that injured tissue or not. That is why isolated tissue has been used for doing the qPCR. And in panel D, what you see is zebrafish Tregs promote organ regeneration by producing tissue-specific regeneration factor. You can see injury, it's spinal cord, heart and retina. What they see is in spinal cord you see NTF3 and IL-10, interleukin 10. NTF3 is in the radial glia, IL-10 is for controlling the inflammation. Whereas in heart, NRG1 is there for cardiomyocytes upregulated, and it can inhibit the inflammatory response also. In retina, IGF-1 is there, which is there in the Mueller glia, and also inhibits the inflammation. So we can see, and the last panel, the E is a model for the alteration of the Treg function during evolution. So what they are showing here is zebrafish Treg, and which has got a extensive regeneration function. In evolution, it has been gone down in the case of mammalian Treg. And when it comes to anti-inflammation, although it is low to start with in zebrafish Treg, and in mammalian Treg, it is quite high. So we can say mammalian Tregs are less of regenerative in nature, whereas zebrafish Tregs are more of regenerative in nature based on their paracrine signaling. And zebrafish Tregs are less of anti-inflammatory in function, whereas mammalian Tregs are more of anti-inflammatory in function. So we should understand when it comes to evolution, the Tregs have evolved in opposite directions. So if you look further, the Tregs in diverse regeneration contexts, Treg depleted zebrafish exhibit compromised myocardial regeneration, forming a thinner myocardial wall with striking fibrin deposition. Remember extracellular matrix, something similar to that of fibrin. Fibrin is a part of the blood clot, fibrin deposition, and persisting collagenous scar. Collagen deposition also is there. So when Tregs are depleted, it favors a collagen depositing environment. Cardiomyocyte proliferation was significantly reduced in the Treg depleted heart, because we have already seen Tregs are associated with proliferating cardiomyocytes. So if FOXP3 is not there, Tregs are not there, hence you don't have too much of proliferation. Treg depleted hearts showed a significant reduction in the expression of specific cardiogenic mitogens such as NRG1, IGF-2A, IGF-2B and PDGF-B. They are depleted. Cardiogenic mitogens. Zebrafish Tregs express all of these factors in the regenerating hearts, but most abundantly they express the neuroglin 1. So Tregs are influential in the proliferation of cardiomyocytes and also in other tissues by doing their paracrine role. Tregs regulate the regeneration of these tissues, which is intriguing because the mechanism by which the spinal cords and retina regenerate are distinct from those of the heart. So we should understand, although Tregs are coming into the vicinity of every damaged tissue, they are not doing the same job in different tissue. So what it tells you, although the paracrine role is there, but in the target cell, it is not causing the same gene expression, because we can understand you don't wear the same kind of dress as you wear in office and same kind of dress on Sunday at the same time at your home, because ambience is different. Same way, these cells can influence those behavior based on which tissue you are influencing. Those genes expression will be triggered. Eventually, the regeneration happens. In zebrafish, spinal cord and retina regeneration is mediated through the de novo neurogenesis from ependymoradial glial cells and also the Muller glia. Zebrafish T-Rex have been shown to promote the proliferation of these precursor cells by dominantly producing neurotropin 3, NTF3 and NT3 in the spinal cord and insulin-like growth factor 1 in the retina. So how contrasting and how different it is. Both are done by the same person. It's almost like you have the same coach, but in one place they teach cricket, another place they teach football. So regenerative factors were upregulated in tissue-specific manner, which suggests the unique capacity of zebrafish T-Rex to produce regeneration factors that are tailor-made, you say, right? Tailored to a specific regenerative context. So T-Rex derived factors. Let us look what are the T-Rex derived factors. One of the major T-Rex derived factors, interleukin 10, IL-10, is a T-Rex derived immunosuppressive cytokine. It is also anti-inflammatory, also keep in mind. Cytokine that promotes tissue repair in mice. Zebrafish T-Rex also express IL-10 in damaged tissue and T-Rex depleted or IL-10 mutant. Keep that in mind. T-Rex can produce IL-10, but IL-10 is mutated, then the T-Rex is not producing IL-10. Mutant zebrafish exhibited an inflammatory cytokine expression profile because IL-10 is anti-inflammatory. Even in embryos, mice embryos, if IL-10 is depleted, they lose their regenerative capacity. So this has been documented. This suggests that IL-10 has a conserved immunomodulatory role in zebrafish T-Rex. However, the proliferation of populations of precursor cells such as cardiomyocytes, ependymoradial glial cells, Muller glia, etc., and the expression of regeneration specific factors such as NRG1, NRF3 and IGF1, they were unaffected in the IL-10 mutant zebrafish. So, which is facilitated and favored by the T-Rex, but which demonstrated that the regenerative response mediated by zebrafish T-Rex is distinct from the well-characterized IL-10 dependent immunomodulatory functions. So, just because, like I gave this example multiple times, if a dish has got 15 ingredients and 16th ingredient is salt, and if salt is missing, those 15 ingredients do not make a food tasty. Same logic you should apply. IL-10, if it is missing, the regeneration can be compromised despite having normal expression of other pro-regenerative factors. So in contrast, the expression levels of the regeneration factor genes were undetectable in T-Rex purified from FOXP3A mutant zebrafish, which showed that FOXP3A is required for zebrafish T-Rex to induce the regeneration factors. So the expression level of the regeneration factor is not seen at all if FOXP3 is mutated, and that T-Rex are almost useless for a regeneration point of view. So the robust regenerative function of zebrafish T-Rex on 3 distinct precursor populations, it suggests that T-Rex play an ancestral and universal role in supporting homeostasis and regeneration of diverse tissues by producing growth factors that significantly enhance the regeneration of tissue-specific cell types. This is very important to note that the T-Rex play tissue-specific roles. Mouse T-Rex can also modulate tissue regeneration by regulating the proliferation and differentiation of tissue-resident stem cells in specific adult tissues, but not powerful enough to bring in a normal regeneration event. So that also tells you while T-Rex are important, T-Rex are not just sufficient to cause a complete regenerative response. However, despite exerting a profound immunomodulatory effect, Tregs are insufficient, not the point, insufficient to promote regeneration in minimally regenerative tissues such as that of mammals. Heart and central nervous system are weakly regenerating in mammalian system, but Tregs are not going to supplement that deficiency or complement that deficiency. Interestingly, although the mice lacking Tregs or FOXP3 function succumb to fatal autoimmune syndrome, zebrafish lacking Treg function appeared to develop only a moderate inflammatory phenotype. There is no autoimmune syndrome as you see in the mammals. This phenotypic discrepancy may be because of the redundancy of the FOXP3B. It has got 3A and 3B. So 3A mutant we are talking. Mammals don't have a 3B. Zebrafish has got a 3B, which was not typically expressed in T cell, but because of the absence of 3A, the 3B expression appears in Treg cells, but could be induced in FOXP3 mutant background through a genetic compensation mechanism when it is absent, although it is not powerful enough to bring about the regenerative response, but it is powerful enough not to have the autoimmune disease as you find in the mammals. So it will be very interesting to analyze the immunological phenotypes of mutant zebrafish lacking both FOXP3A and FOX3B mutant in zebrafish to understand the effects of FOXP3 gene duplication on anti-inflammatory regulations in zebrafish. Alternatively, we can also do the moderate inflammatory phenotype observed with zebrafish that is lacking Tregs might also suggest that the attenuation of the immune response in aquatic animals, how it has evolved, that will give a good insight into those angles as well. Mammalian Tregs might have lost or have limited pro-regenerative capacity at the expense of gaining a robust immunosuppressive function to harness rapid and strong inflammatory response that has evolved to seal wounds. If there is an injury, forget about regeneration, seal the wounds with impermeable scars in terrestrial animals because they are more vulnerable for wear and tear. There is no water support cushion around their body. They are more vulnerable for dryness and more injury. So dry environment can favor more of damage to the body because there is no cushioning water around them. So a solid scar is favored. Like zebrafish, neonatal mice can regenerate damaged heart tissue through cardiomyocyte proliferation through its self-renewal capacity quickly diminishes after birth. One recent study have revealed that the regeneration of neonatal mouse hearts is also facilitated by infiltrating Tregs through the modulation of inflammatory macrophages and the production of growth factors that promote cardiomyocyte proliferation. So the factors produced by the neonatal Tregs includes amphiregulin AREG, which is required for the Treg mediated lung and skeletal muscle repair. So the host or the local tissue support in the form of a gene expression favors Tregs function. Zebrafish Tregs also express AREG in a non-tissue specific regenerative context. Zebrafish also have got the same gene. Thus, the paracrine role of Tregs for cardiomyocyte proliferation is conserved regenerative mechanism between zebrafish heart and the neonatal mouse heart. However, it is still important to determine whether the mouse Tregs require tissue-specific secretory phenotype in regenerating hearts. It is also important to note that mouse in vivo expansion of Tregs can reverse the autoimmune dysfunction. These studies suggest that genetically modified Tregs could serve as an ideal natural vector for delivery of tissue and disease-specific regeneration factors that will activate the latent regenerative pathway. We will study more about regeneration in the subsequent classes. Thank you.